Hi, so today on this episode of Root Docs, which is a mini series dedicated exclusively to the discussion of root diseases, our guest today is Dr. Maria Tomaso-Petersen, Professor Emeritus at Mississippi State University. And today we're going to spend some time talking about a disease that I think Maria is somewhat familiar with, and that is take-all root rot and Bermudagrass decline. So, Maria, thank you for being on today's show.
Well, Jesse, thank you for inviting me, and I'm looking forward to our conversation.
Yeah, this is really exciting to me for a couple of reasons. And so, when I left the University of Tennessee, I was somewhat in tune with what you all were doing, with these Bermudagrasses, and then, I needed a job after graduate school, moved to Chicago, and then after that I moved to Canada, and now I'm back in the Southeast, and I kept on reading all of the work all of you have done with some of these diseases, and it is really, really impressive. And my first question to you, for me is, we hear take-all root rot, and then we hear Bermudagrass decline. Are those the same thing, or are those different?
Well, that's a really good question. And I like to go back to the beginning. And the beginning in 1984, the disease was characterized in an experiment station bulletin in Florida, and it was called Bermudagrass decline. But the symptoms they described were circular patches of chlorotic Bermudagrass appearing late in the summer. And then in 91, Monica Elliott characterized Bermudagrass decline, the disease, and identified the causal agent as Gaeumannomyces graminis. Then, as she continued to work with that fungus in warm season grasses, they found the same pathogen, Gaeumannomyces graminis, or GGG is what we called it, infecting St. Augustine grass, causing chlorotic patches, and she called that take-all root rot. And then those warm season grasses that weren't a Bermudagrass green were referred to, that disease was referred to take-all root rot. And then in the early 2000s, I was doing diagnostics, and I identified these chlorotic patches in the spring of the year on Bermudagrass greens, champion ultra dwarfs, and it was loaded with the pathogen GGG, and I referred to it as GGG patch because when I was coming through the literature and learning about this disease, the decline was more found on the edge of the greens. It looked like it was worn out, and it just kind of faded. The turf faded and lost density, and it wasn't so much circular patches. Yeah. And then okay, let's see, about maybe 10 years later, or it was in the late 20-teens, Jim Kern started to see this patch disease on their ultra-dwarf Bermudagrasses, and it was just blowing up in the Carolinas. And he called it and referred to it as take-all root rot. And so we've kind of just gone back and forth and kind of just melded the name. It's a synonymous name now. And in the Compendium, it's referred to as Bermudagrass decline, but it also says, this disease can also be known as take-all root rot. So I think the take-all root rot is probably taking hold, and it better describes the disease and what's happening to the root system than like decline. It's doesn't really have to be a pathogenic problem when we see different decline type names for diseases. So I think in the future it'll pretty much be referred to as take all root rot, or TARR as the
Yeah.
abbreviation would be.
Yeah. Yeah. Because that, you think about, this train of, you mentioned St. Augustine gets it, and, all the work being done on some of these new zoysia grasses being, built for putting greens now. It would be kind of odd to call, on a zoysia green, oh, that's Bermudagrass decline on
Right.
zoysia.
Yeah, yeah. So,
And so on say Bermudagrass, so it the symptoms of this doesn't have to be that kind of patch structure that we see, in the kind of take all root rot in late summer, fall. But you're seeing this pathogen cause that kind of general thinning and more diffuse symptoms as
correct.
well.
Yes. And in the deep south, my experience and through diagnostics, it was more of that thinning, declining that you would see, and it usually started around the edge of the green where the cleanup layer in mowing would occur, and that's just more reduction in plant height, and those thinning out symptoms are associated with low mowing height, and
Yeah.
so that's what I was more accustomed to seeing. And, but now in our area and in the deep south, they're starting to see a lot of those patches develop in the fall. And then as the grass greens up in the spring, those are the weakened areas. So they look patchy too. So
Yeah. Yep. And
it's interesting.
reading some of your work, it was referenced. You just mentioned Elliot, and what they found, or what they suspected, is that there's multiple pathogens likely present, and it's almost like take all root rot, almost like a general term, kind of like snow mold, where there's multiple pathogens that could be causing very similar symptoms.
Correct.
It How many have you guys identified as being in that complex that are causing those kind of take all root rot or Bermudagrass decline symptoms?
In our research, my student Philip Vines did that work, and he actually identified, I think it was 6 species that were
Mm-hmm.
not Gaeumannomyces graminis, that when we did the phylogenetics, they didn't match anybody. But through the pathogenicity tests, we concentrated on those that showed pathogenicity to the roots. And through that, we've identified 3 species that were pathogenic. One was a new genus species, Candidacolonium cynodontis. And then there was a Magnaporthiopsis species, which was kind of interesting because it used to be called Magnaporthi. And back in the day, Magnaporthiopsis was the summer patch fungus. And I was like, why is there Magnaporthi species on this warm season grass? So that was like in the early 2000s. When I just started tinkering with it through diagnostics, I would see a lot of the gross cessation structures on some roots, and I didn't really associate that with the Gaeumannomyces species more so than the Magnaporthi species. And then I started to see different shaped hyphopodia, not just simple and the deeply lobed, but different size lobed hyphopodia, some that were more kind of mitten shaped, and I was like, why are these hyphopodia different? Is it the age of the fungus when it's infecting and I'm seeing it, or what's the
Yeah.
deal? So, I put all those questions on the back burner until the mid 20 teens, like 2012 is when Philip came in, and we talked about maybe let's see what these are and who they are. The phylogenetics and the molecular
Yeah.
work was getting so much better. And back in the early days, I took a lot of these ectotrophic root infecting fungi that I didn't think was Gaeumannomyces graminis. And back in the day when I did ITS blasts, excuse me, I'd get like 96% identity to Magnaporthiopsis. And I'm like, well, who is this fungus then, you know? And what's it doing on the warm season grass?
Who are you and what are you doing here?
So I was, you know, through his excellent work, we were finally able to, you know, go into that black hole of these ERI pathogens and find out exactly who is there. And Elliott and Lanscoot, you know, discussed there's a complex. And so we were finally able to, you know, solve that riddle and identify these fungi. And then over in North Carolina, Jim Kern's student, Cameron Stevens, he found and identified, which was already identified, Gaeumannomyces graminicola, which we never found in our work, which I thought was interesting because, you know, we had hundreds of samples and putting green samples from Hawaii all the way through the deep south and everywhere. Yeah. And so I was like, well, why didn't we come across graminicola? You know, that's just kind of interesting. And which Cameron showed was highly pathogenic. So, and then we would do samples and we would find multiple species on these rotted roots together. So, you know, that was pretty interesting. So, yeah.
Yeah, I could imagine. And, you know, you got the craziest thing is that they're somewhat related in their behavior, but from a taxonomy perspective, they're not even in the same genus. Like you mentioned a couple of these that aren't even, and so these are very different organisms, which, you know, thinking as a superintendent, you tend to think, okay, I got one disease caused by this pathogen, and I know what this pathogen is very sensitive to, say from a fungicide perspective.
Correct.
But that takes a lot of work because now you have to almost go back to the drawing board and think, okay, we've identified, I mean, the very different organisms all infecting these stolons and roots at the same time. Right. And how are they different? Do they vary in fungicide sensitivity? Do they slightly vary in temperature? Because you've also found that too, differences in fungicide sensitivity across these various modes of action.
Correct. Yes. And different temperatures as well. We did temperature studies as well as Cameron, and I thought it was really cool that, you know, NC State was doing research with these fungi, and they were finding the Magnaporthiopsis cynodontis and Candidacolonium cynodontis as well. And then there's Gaeumannomyces nanograminus, a new species. So they were finding all of the species we were finding as well as their graminicola. And different temperature, optimal temperatures, and they responded differently to fungicide modes of action. So it made me start thinking, as we worked with spring dead spot and found that there were different species, and we knew that in the beginning, but the work that was done at NC State by Lane Treadway when he was still there with fertilizers, it's almost like the work is going to have to continue with these fungi to see in greens, okay, who's the predominant species? Because there might be a predominant species. And also, you know, is the reaction to the fungicide different, or can we kind of do a blanket applications and see positive results with the fungicides?
Yeah. And last night, I want to show this because last night I was doing some kind of background reading on this subject, and you know, there's always some people, you know, there's no such thing, because you've spent some time under a diagnostic microscope. There's really no such thing as a sterile root zone sample that you can just about find. It's So I'm actually going to share my screen, and this is clipped from, I think, the thesis from Tucker that he did.
Okay.
And I want to see if I can find where that screen is. Right here. I found this hilarious. So this is from I believe the master's thesis, where you guys sampled the crap out of some of these greens. This is an outline of a putting green that was sampled in 2 consecutive years. And you can just see how many sampling points. Credit to him. That is a ton of work. It was fascinating. There was only one tiny sample in both years that you guys weren't able to detect one of the take-all root rot pathogens present. Is that a kind of a good way to interpret this?
Well, what we saw was the dynamic change from year to year on who was colonizing those roots. And, and yeah, those plots were small in, 2.4 meter area.
Yep.
And the fact that the colonization was not consistent from year to year just goes to show you, the diversity that's out there, and
Yep.
the vigor of those fungi in that root system. And each year those roots kind of slough off, and so there's a new flush of root, and it's like, okay, whoever's there from one year to the next gets to get on those roots and colonize. Yep. Whether they do damage or not, they're still there. And this research that Aaron Tucker did it was outstanding, and it just kind of blew my mind to the point where, how do you wrap your head around this kind of stuff, to make it into a publication that kind of makes sense. And if I was a superintendent, I'd kind of be intimidated and afraid at what we found on this, putting green out at our MSU golf course. So that stuff is going on. Give credit to the superintendents that they have grass and it's growing in the presence of these fungi, because, it is a stress-related disease, in that if there's any kind of a physiological stress, I think the pathogen's going to get the upper hand. But by maintaining that healthy turf, you can sample all you want, and you're going to find these fungi day in and day out on healthy plants. But it tells you, be sure to keep them healthy, because the chance of those roots declining to the point where that grass can't recover and generate more root system to support it, it's very fragile.
Yep. Yeah. So it's everywhere. And then in some cases, you got all 4 of these pathogens present, in that same sample, or on the same root or same rhizome. That has to be troubling. And then as you said, something happens to where you get this infection, and then it seems like there's this latency period or delay of when you actually see the symptoms. And we think you guys think it's plant stress related, that it's getting colder, the days are getting shorter, and then all of a sudden it gets flipped, and then we start seeing those symptoms.
Yeah, that weakened turf. Yep.
So that would make it challenging as a superintendent to chase this down from a curative perspective, as far as, so I got these symptoms, and let's just pretend it's, say, mid-September, October, you got these symptoms. That this would be very hard to chase down from a curative perspective, because you're going to need the plant to grow out of all of those symptoms during a time when the plant may not be growing that fast to begin with.
Absolutely. And, we see that with Mini-Ring as well. And so it's just like with spring dead spot, you have to kind of map those areas. If it's the entire green or areas within that green, and know where those affected areas are. And then that next growing season, you have to be on top of it. And, stay in tune with the soil temperatures, and get your fungicides out according to those soil temperatures. And that's another thing Cameron Stevens did a great job on, and I know Jim Kearns is continuing to do management using timing and relating it to soil temperatures. So I think there's good news coming, and he's identified fungicides that really help reduce that disease symptom expression. So I think there's hope for it, and just be on the lookout for that. I did more of the biology of the fungi and not as much of the management with fungicides.
Yep.
In our work, so.
And for soil temperatures, based on, you're working in what they've done at NC State, soil temperatures, when would you, if I'm a superintendent somewhere in the Southeast managing ultra-dwarf Bermudagrass putting greens, what do you think would be kind of that preventative application time frame, as far as what soil temperatures do you think you would look at, and what range of soil temperatures would you pull the trigger on a fungicide application?
I would look at the soil temperatures when they start getting into the 80-degree Fahrenheit, 80 to 86-degree Fahrenheit, which is similar to when Bermudagrass roots are growing. And target those areas. And in Mississippi, we kind of got the soil temperatures and then went across the growing season. And we were in May.
Yep.
And so for our area, the Deep South, you might target May. As you go into the Carolinas, you might, target mid-May, depending on where you're located. In Texas, they'll start probably in April with those warmer temperatures, the end of April. So that's kind of a good target. And so many of our fungicide applications now are kind of associated with soil temperatures, which the superintendents are getting used to looking at soil temperatures and not a calendar date, because, our crazy weather, for large patch, you could be putting out fungicide for your spring application, in March instead of April or May down here. Yep. So I think they're getting, used to looking at the soil temperatures. And I think that's a good key, is to look at, okay, my first application should be in the 80s to low 80s. And it also will help protect that root that's being developed and starting to grow. And then, of course, follow-up applications on I don't know what's recommended, maybe 21-day, 28-day repeat application.
And then at what temperature do you think you're safe? So let's just pretend, okay, we're hitting that 80 degree mark in May. What is the lower end of that spectrum, do you think, where, okay, the soil temperatures have dropped to this point, I can breathe a small sigh of relief? What range do you think that would be?
Well, we saw fungi cease growth at like in the low 60s, for these particular fungi. And it's interesting because then the take-all fungi that affect cool season grasses, well, they're growing at 65. But our warm season fungi, they kind of cease growth in that 65 degrees. So I'd say below 70, they're going to start slowing down and just hanging out and not being infective like they would at their warmer temperatures.
Yep. That makes sense.
Yeah.
for me, I was reading the Compendium, which you were a co-author of this, and what was I kind of just staying on this Compendium for a second? What was that like being involved in that publication? Which in the industry we kind of call that the Bible, so to speak, of turf pathology. It's one resource that I know for me, it is usually right next to me or in my backpack when I'm traveling.
Absolutely. I used that book as my course book for my class, Turfgrass Diseases. But to work on it, it was very it was fun for me. And the way I approached it is I just went back to the beginning and did research on, the first mention of this disease or any of the diseases I worked with, and then went through and looked at the fungi that were associated with it. And from our third edition to the fourth edition, so many of the taxonomy, had advanced to the point where a lot of the fungi were called different names. And so it was kind of fun to go back and just, clear a path and write about the progression of the disease and bring it up to date. It was very satisfying. And for me, it was kind of the end of my career. And so it was kind of like a capstone for me to work on this and put in a lot of information on warm season grasses that the other compendia lacked previously. So we're pretty excited about it. The editors, when we finished, we were like, man, it's good for at least 10, 15 years.
It's such a good resource. And I definitely see a lot of the southern diseases definitely bulked up in this version. If you haven't gotten that new compendium, I would, especially if you're managing ultra dwarfs in the south. My next question, just out of my own curiosity, take all root rot Bermudagrass decline is one of the big diseases of ultra dwarf Bermudagrass putting greens. So is Mini-Ring. Have you ever seen symptoms of Mini-Ring and symptoms of take all root rot on the same green at the same time?
I have not. That's a good question, but I have not.
I have not either, because I was reading, some of the cultural management strategies, and Joe Roberts talks about this at all quite a bit when he's talking about Mini-Ring, is if you have Mini-Ring, do not put ammonium sulfate out.
Correct.
You don't want to put ammonium sulfate out. But for one of the cultural management practices for managing take-all root rot, it says to apply ammonium sulfate because that can help reduce it's almost like a damned if you do, damned if you don't. So I'm going to put down ammonium sulfate to help limit my take-all root rot. But I wonder if then that open the door for Mini-Ring to come on in?
Well, that's interesting because it's almost opposite. The cultural practices for the take-all root rot decline, cultivation, core aerification. I've seen greens where in the thinning aspect of that disease where they core aerified, the only place that they had grass was in those little cores,
Mm-hmm.
But for Mini-Ring, any aggressive and like continual cultivation of aerification, top dressing, a little deep, promotes that disease. So you are in a catch-22. And again, I think it goes to proper fertility management in the beginning with sound, cultural practices. And preventative fungicides and get him out there. So,
and so talking about putting it all together. Take all root rot, Mini-Ring. You got for a superintendent, who say used to be managing bent is now switched over to Bermudagrasses. Man, these Bermudagrasses have a lot going on, and And what you're saying, Maria, is Bermuda grasses absolutely do get disease. They are not just these immune grasses.
Yeah. It's not the old 328
Yeah.
at a quarter inch anymore. And absolutely, they're high maintenance, high disease maintenance grasses. And we started seeing that early on in the late 90s when people were starting to, put out Ultradorf a little bit more. And, they started planting that grass and we really didn't know anything about it.
Mm-hmm.
And my former major professor and boss, Jeff Kranz, we would talk and he was like, there's so much we don't know about these grasses. And look at how much we learned about the thatch management where we didn't have that with the old Ultradorfs. And so it opened up a whole new world. And then the recovery is slower in disease recovery. And I think, that high maintenance, high management, short mowing height just lends to more disease.
Yeah. Yep. And, not to make this even more confusing for a superintendent, who may be watching this. So you got multiple pathogens causing take all root rot that they form this kind of complex. Do you think the pathogens that cause take all root rot symptoms are also potentially forming a complex with other pathogens of different diseases, such as Pythium species or perhaps a root knot nematode? Do you think they're all kind of tag teaming the plant and just kind of compounding the problem?
Well, I think it's a cause and effect type thing with a compromised root system due to the take-all pathogens, and the Pythium's always there. And we know that Pythium and the nematodes are somewhat associated and are found together a lot. And, that whole microbiome of pathogens affecting that root system, I think weakens, opens the door for other pathogens. And, it could be a cascade effect, but it shows what great management our superintendents are doing on these grasses, because for the most part, their greens are, looking good and satisfactory. And so, it's high stress.
Yeah.
Doing disease management. And then you've got the foliar diseases you've got to worry about too.
Yeah. I'm actually going to finish up, even though, this is Root Docs, which is discussing, root pathogens. And you mentioned when you were, looking at all these ectotrophic runner hyphae and all these different pathogens causing, take all root rot, that they weren't exactly looking the same. The hypophodia is a little different. That curiosity got to you when it came to discovering another new pathogen that your lab helped work on. And that is the pathogen that causes Inkspot, correct?
Correct.
What was that like from the first time you got a sample and you're looking at it? And I'm just trying to imagine, you're looking at a sample and you think, okay, I think this is a curvularia, which could take sometimes less than 10 seconds to confirm under a scope. And that didn't happen with that one. So could you kind of walk through the birth of Inkspot, if you will?
Okay. Well, it was a Friday night in the lab, and it was actually a 328 sample, and it had foliar symptoms that just didn't look like a dollar spot symptom, and it didn't look like a bipolaris leaf spot symptom. And I was like, what in the world is this? So I immediately remove some of the infected tissue, and we surface disinfest, and I just put it on water auger. And I thought I'm going to let it grow out over the weekend and see what's there. But in the meantime, when I was looking at the plant, I saw a lot of very black hyphae down kind of in the stem region of the canopy. Mm-hmm. And I didn't see any sporulation or, sexual reproduction going on like you would if it was Curvularia lunata, mm-hmm. which, sporulates profusely. And I didn't see any of that. And I thought, well, this is pretty weird. So the next Monday I came back and there was this robust hyphae coming out, and it was clear and there was no sporulation, but within a day or two, it turned black. And I was like, what in the world is this? I had no clue. And I had to report to the superintendent, there's a foliar disease, but I don't know what this is. I've never seen this before. And so that was in 2007. And 2016, we were finally able to publish after years of characterizing the fungus and figuring out, where this fungus belonged in a genus. And it kept going into Curvularia. And I thought, well, it's just not sporulating. And I'd put it on multiple nutrient augers and put it in light, UV light, no light, 6 months in the dark. And it just never sporulated. Nothing. So the taxonomists, when we were publishing, they were very apprehensive because they said, well, it's Curvularia and it's not, sporulating. And there was a book, The Atlas of Turfgrass Diseases.
Yes. Yep.
And it came out of Japan. Mm-hmm. And there is a Curvularia blight that they have a chapter of. And the pictures are exactly like the symptoms that we were seeing here. And I first saw it in Texas. And over the years, then I was seeing it on our research screens at Mississippi State. And I was very excited about that. But in this book, the causal agents were Curvularia lunata and several other sporulating Curvularias. And then a little mention down below, and a sterile fungus was associated with it.
Yeah.
And I was like, I've got the sterile fungus. And I showed that it's, much more pathogenic than Curvularia lunata, and it, produced the same, symptoms. And so, it took a long time and, trying to break through and, show that, okay, this is a Curvularia, but it's sterile. I don't know why it's sterile, but, in some of the old writings of like, Dreschler, when he was characterizing the Helminthosporium fungi, he talked about some are just sterile. Yep. And, you just have to accept it. And this unique fungus, which is highly pathogenic, and I think the most pathogenic Curvularia in the group that are associated with diseases, our turf diseases, this is highly pathogenic, and it causes some pretty major damage in the foliar region of our greens. It's on fairways, it's on tee boxes. I haven't seen it in athletic fields or home lawns, just, shorter mowed golf turf.
Yep.
It likes a lot of water.
I happened to visit Dr. Joe Roberts at his research station in Florence, and he is convinced he has it too. And that damage, I remember thinking, holy cow, this thing is aggressive. It's fascinating to me, those listening to this, it may have kind of skipped over in their minds how much dedication that took on your end to keep working on this thing. From that day you first saw this to the day it was published was almost 10 years. That is a ton of work, a ton of dedication. And
Yeah, I was pretty happy to see it in print.
and that dedication is why you are absolutely one of the best turf pathologists we've ever had. We're very grateful for all that you've done, and really appreciate you being on this show. And you can find this episode on our NView website and on YouTube. So thanks, Maria, for being on the Root Docs miniseries.
This is awesome. Thank you so much, Jesse, for your invitation. I enjoyed it.